Hello everyone, I am Divya from Spire Lab Electrical Engineering Department, Indian Institute of Science, Bangalore, India. Here I will be presenting our paper titled Automatic Glottis Detection and Segmentation in Stroboscopic Videos Using Convolutional Networks. These are the contents that I am going to discuss in this presentation. First, I will talk about the motivation for this work in the introduction. As we know, voice is produced by the vibration of the vocal folds. It is essential to study its vibration patterns to diagnose the voice disorders. And also, the diagnosis is commonly based on the visual inspection of the vocal fold vibration, which is a challenging and time consuming task in general. So, this brings the need of automatic glottis segmentation methods in the diagnosis of voice disorders. Here is the image that has been taken from a video stroboscopy recording. This is epiglottis and these two are the vocal folds located inside the larynx and the space between the two vocal folds is the glottis. This figure illustrates the healthy versus pathological vocal folds. In the pathological case opening phase, we can see a groove kind of structure in the vocal fold which leads to an incomplete glottic closure during phonation. This type of vocal fold condition is called sulcus vocalis in voice disorders. This figure shows the sample stroboscopy video image from all the 22 subjects used in this work. From the figure, we can observe a high variability among the glottal shapes of different subjects. We can also observe a low illumination in some of the images. And it is clear from the figure that the camera position relative to glottis changes across all the subjects. This figure illustrates the example images to show the supraglottic structures covering the glottis region. Here, images have been obstructed by the epiglottis, which makes the glottis invisible in all the cases. Due to this, glottis position varies from one image to another image, which is again a challenge to glottis detection and segmentation algorithm. To make the predictions robust to this kind of translations, we use glottis detection network prior to the glottis segmentation. These are the few algorithms that are proposed for automatic glottis segmentation. In deep neural network based approach, they used RGB values from a 3x3 neighborhood of a pixel as a feature vector to classify whether pixel belongs to inside or outside the glottis region. But it performs low in the frames where there is more disagreement across the annotations. In fully convolutional network based approach, they use a bounding box to detect the region of interest followed by the FCN for the segmentation. In the region growing approach, active shape model is used to find the glottis boundary, but it fails in unseen subject cases when the glottal shape is much different from those in the training set. In this work, we address the problem of glottis segmentation using a type of fully convolutional network called UNIT. It has shown very good results in biomedical image segmentation. We'll explain about the UNIT in further slides. The goal of this work is to propose an automated method for glottis detection and segmentation in stroboscopic videos using convolutional networks, and also to study the performance of unit with and without skip connection using three different weight insulation schemes followed by the fine tuning to achieve accurate glottis segmentation. For experiments, we consider stroboscopic videos from 22 patients with sulcus vocalis data in this work, out of which 14 are male and 8 are female subjects. And all the videos are recorded using Xeon Endostrobe E with 70-degree rigid scope. And each video has a frame rate of 25 frames per second with a frame resolution of 720 x 576. The manual annotation for glottis detection is done for 24,970 images which are extracted from 22 stroboscopic videos. And labeling is done by manually segregating the glottal and non-glottal images into two groups. Out of 24,970 images, only 921 randomly selected images from 18 subjects are used for glottis segmentation. Here, a MATLAB-based graphical user interface is developed to mark the boundaries of the glottis, and each image is annotated by three speech language pathologists. This figure illustrates the three example images and corresponding annotations by the three SLPs to show the inter-SLP agreement. And mean die-score across pair of SLPs annotations is computed and it is provided at the bottom of the respective column. We observe that the annotations often vary across SLPs, particularly when the glottal opening is small as shown in the first column of the figure. In this case, mean die-score is low, which indicates a low agreement between the annotators. The proposed approach mainly involves two steps. One is glottis detection, in which for a given sequence of frames detect the frames with the glottis. And another one is glottis segmentation, where for a given glottis image segment the glottis. We have used a fully convolutional network based classifier for glottis detection, where it classifies the presence or absence of glottis in each frame of the video. We have used 24,970 frames for glottis detection, and we consider images with clear visible glottis as one class and remaining all images as other class. We denote this network by GDN. Coming to the architecture, this network consists of seven main blocks, where each block consists of convolution, batch normalization, and relay activation layers followed by the max pooling of 2 cross 2. Finally, we have used a sigma layer to classify if image contains glottis or not. This figure illustrates the block diagram of the proposed approach for glottis segmentation. In this work, we have only 921 annotated images to perform the glottis segmentation task, which are significantly less compared to the number of parameters of the unit. To overcome this problem, we have used the trained GDN weights for better insulation in unit to get more precise segmented output compared to random initialization. In order to reconstruct a binary image from the predictions, we choose a threshold of maximum of predictions minus 0.05. Coming to the unit architecture, it consists of a contracting path with two encoders and an expansive path with two decoders. Each encoder consists of a convolutional layers with relay activation followed by the maxi pooling. Similarly, each decoder consists of an upsampling layer followed by a convolution and relay activation. At the end, a convolutional layer followed by sigma activation is used to classify whether each pixel of the image belongs to inside or outside the glottis region. Here, a concatenation layer or skip connection is added in the second decoder after an upsampling layer with the correspondingly cropped feature map from the contracting path to the expansive path, which is called as unit with skip connection or USC. And its absence is termed as unit without skip connection or UWSC. Now let us see the experimental setup for JDM. For glottis detection, we have used 24,970 frames from 22 subjects with a image resolution of 224 cross 224 cross 3. We have used four-fold cross validation setup for both detection as well as segmentation tasks, and fold structure is shown in the table. The network is trained using a binary cross entropy loss function. Here is the experimental setup for glottis segmentation network. We have used 921 images from 18 subjects to train the unit. As there are no labels for the subjects S5, S11 and S11, S17 and S22, they are excluded from the corresponding folds for the segmentation task. We use weighted binary cross entropy loss function and Adam optimizer to minimize the loss. And unit is trained for a maximum of 70 epochs with early stopping criteria by monitoring validation loss. We consider DNN-based approach as baseline in this work. Both baseline and the proposal method are evaluated with the same dataset and fold structure. We have used classification accuracy and localization accuracy and die-score as an evaluation metrics in this work, where classification accuracy is used for glottis detection and other two for glottis segmentation. Here, die-score computes the overlap region between the predicted and the ground truth pixels, and it is calculated using the given formula. A total of 6 experiments are done with 3 weight insulation schemes using 2 architectures, which are unit without skip connection or UWGSC and unit with skip connection or USC. The details about these networks are explained earlier. The different weight insulation schemes are as follows. One is random weight insulation, where the weights are initialized randomly. Second one is detection network weight initialization, in which GDN weights are used to initialize unit encoder weights. And third one is detection network encoder frozen weight initialization, where the weight initialization is done in a manner similar to that of the DNA, except that encoder network weights are not updated during time. This table illustrates the average classification accuracy and corresponding standard deviation in brackets, which computes across all the 4 folds on test and validation data for glottis detection network. Here the performance of GDN is affected by the images which have small glottis or supraglottis structures that covers the glottis region, which leads to mixed classification of glottis and non-glottis in both the cases. This table illustrates the mean die-score and the standard deviation across 4 folds on test and validation data for all 6 experiments. Among all USC DNA or unit with skip connection, detection network weight insulation has shown the best performance. and it is considered as our proposed approach. The table shows the fold-wise localization accuracy or die score across three SLPs using baseline and proposed method. The three values in each cell indicates the localization accuracy or die score calculated with respect to the three SLPs annotations. Here, die score is computed with and without detection network to achieve good results. The die score computed from the proposed approach and baseline is considered as die score without detection. And in case of die score with detection, it is computed for USC on the frames where the glottis has been detected by the GDN. These are the observations from the table. It is observed that the localization accuracy and die score are improved for all the annotators using the proposed scheme compared to baseline. And both localization accuracy and die score with and without detection are high for fold 1. This is due to the clear visible glottis image presence in the subjects S1, S2, S3, S4 and S5. And also for fold 2 has low localization accuracy among all for both the tasks. This is because of poor illumination in the subjects S6, S7 and S9. It is observed that the localization accuracy for fold 4 is high. But the die score obtained with and without detection network is not very high compared to all other folds. This could be due to the supraglottis structures that covers the glottis openings. Interestingly, the die score without detection is high compared to the die score with detection, which shows that the performance of glottis segmentation network is increased by using glottis detection network weight initialization, although it's fine-tuned with the less labeled data. And low variance in the average localization accuracy and die score across three SLPs shows that the robustness of the proposed method. We got an average localization accuracy of 81.3% and a die score of 0.73 from the proposed approach, which outperformed the baseline scheme by 15.87% and 0.07 respectively. This figure illustrates the cases where the proposed method performs better than the DNN-based baseline approach. In this figure, column A contains three sample images with corresponding subject number and die score achieved by the proposed approach and mean die score across pair of SLPs annotation. Column B, column C, column D are corresponding ground truth and segmented image by baseline and proposed method respectively. From the figure, it is observed that the proposed method performs better than the baseline even when the images are affected by the poor illumination in all the cases. And also it performs better even when the glottis opening is small, as seen in the subject 14. While it performs uh, low in the case of baseline approach. The proposed method is not performing well for the image in the last row where the glottis opening is blocked by the supraglottis structure. With this, I am going to conclude our work. It is observed that the localization accuracy and die score are improved for the all annotators using proposed scheme compared to baseline. And the performance of glottis segmentation network is increased by using glottis detection network weight insulation and the skip connection used in the unit. As part of future work, we want to improve the performance of unit using different loss functions. Furthermore, we would like to extend this algorithm for all types of voice disorders. We would like to thank ICE Research Fund for their support in this work. If you have any questions or suggestions, please write to us at spirelab.ee@iasc.ac.in. Thank you.